I think this leaves an opening to where we are, to where we're going. And basically, for the first time in a long time, we have drugs that were developed and approved specifically for the treatment of all migraine. And these are the CGRP drugs. And the FDA has now approved 3 monoclonal antibodies for the treatment of migraine. And these are antibodies directed against calcitonin, gene-related peptide, or CGRP. There are now 4 monoclonal antibodies that have been approved or will be approved for the treatment of all migraine in adults. We have galcanezumab, erenumab, which we talked about. Galcanezumab is an antibody against CGRP, whereas erenumab, the CGRP receptor. Fremanezumab and apinzumab are both antibodies against CGRP itself. So we have 3 antibodies to CGRP and 1 antibody to what Peter talked about earlier is the canonical CGRP receptor. Peter, what's the rationale for targeting CGRP in migraine? I think you had something to do with that, didn't you?
Well, the rationale began as an exercise in trying to understand which of the trigeminal neuropeptide transmitters were important in migraine and whether one could target them to stop migraine. So, and I think there are three strings to this to that bow. Lars Edmondson and I did a study that showed that you could during a very bad migraine in the pre-triptan era, there was elevated CGRP and not substance P in the cranial circulation. And then when the triptans came along, we showed you could reverse that with a triptan. And then Jess Olesen, some years later, used CGRP to trigger migraine in migraine patients. And the fact that it was elevated, you could reduce it, and then it would trigger a migraine attack. Those 3 things, I think, went together to say to people interested in developing medicines that CGRP was a target.
So we all owe this to you, don't we?
Oh no, everything's of its time. Penfield, McNaughton, and Bill Findell had done incredibly good work showing that the crucial innovation, pain-producing innovation within the head was in the dura mater, in the first division. What we did was build on that by activating trigeminal dural afferents and build on work that people had done that had been done to identify CGRP in the trigeminal ganglia that Lars had done, and put the 2 things together. And then probably the crucial thing we did was translate that into humans, take that experimental work and ask the question in humans, if you tried to do that and you tried to block it, what would happen? Everything, you build on the shoulders of giants, they say.
We're glad you did, Peter.
One of the questions that I'm interested in, as we all are, I'm sure, is both antibodies block the CGRP pathway. If you don't respond to a receptor-targeted antibody, is it because a lot of people have waited for these drugs for a very long time, and they put all their eggs in the CGRP basket, so to speak. And so you can imagine how devastating it would be to start on one of these antibodies and not respond. Like, what's next after I've been waiting for these for 6 years? And so one of the questions that's going to be important to answer is, if you don't respond to a peptide targeted antibody, will you respond to one that targets the receptor, or vice versa? And so we're going to have fun sort of dissecting that apart.
Anybody have any preliminary data?
Well, I would like to say about the receptor, it might be worth saying in terms of the receptor, why are we using monoclonal antibodies? So sometimes colleagues will ask us that. The receptor system that's involved is a combination of a 7-transmembrane G protein coupled receptor, like a classic beta receptor, and a protein that sits next to it called the receptor activity modifying protein, or the RAMP, and there are 3 of those. And if a particular, the calcitonin-like receptor sits with RAMP1 on the cell surface, that's the CGRP receptor. Sometimes people will hear the term canonical CGRP receptor, and that's the one that erenumab binds to. Now already that's a complex structure. some years ago, there were small molecule CGRP receptor antagonists that were developed, and they had their problems with metabolites causing liver enzyme problems. The reason we've ended up with monoclonal antibodies is it's been difficult to develop these receptor blockers, so that there's a certain logic to how we've come to where we are. And I think it's sometimes useful for colleagues to understand why we suddenly jumped there.
Getting back to your earlier question about, do we have any early clinical data about if you don't respond to the receptor binding, do you respond? We have seen some patients make the transition over, and they are responding to different items. So those that were on the arenumab that was released earlier, who then transferred over to galcanezumab or fremunizumab, and are starting to see responses when they weren't seeing them before. That's very early, very small number, but we are seeing that.
And the reason I bring it up, of course, is that I think it's important for neurologists to be able to counsel and speak to their patients and say that, there may be reason to believe that you may respond to one even if you've not responded to the other, even if they target the same pathway. We've seen this with triptans, right? Yeah. They're all 1B, 1D agonists.
And partially F.
And partially F, a few of them, but nevertheless, they all sort of target the same receptors. So why would you respond to one and not the other? Yet we have 25 years experience that tells us that that happens all the time.
Well, another interesting question is if you respond to one, CGRP binding, ligand binding drug, would you not respond, or would you respond to the other? And if you have an adverse reaction to one, would you have to another? Is the adverse reaction really related to the excipient, and the excipients are different in each one of these, or is it really related to the monoclonal antibody itself? And I think this is going to be an amazing time to learn about this, because I agree. I think there's a similarity to the triptans, and sometimes the reactions that people have to the product that makes it doesn't work because I'm not tolerating it, and just making the switch, they tolerate it better, and now it's working well.